to see through them. But there's some other labs we do. You probably going to want goggles. And they're over here, our safety goggles. So today we're going to be looking at compound microscope. And let me see, I'm going to have some stuff to pass out. So did you guys download the lab report template from the class page on Gradely?
Yes, sir.
No problem there, right? Good. Okay. And what they want me to do is hand out an example. The lab we did last week was the whirlybird. Okay. And this is an example, I think, of a write-up. And I'm going to add one thing to it that they didn't include in here. Okay. They didn't show their calculations. You see the data in the table. They didn't show the calculations. You do not need to type up a lab report. In reality, if you're doing a lab, you're sitting down, you're looking at something through your microscope, or you're dissecting something. You're not trying to type it up at the same time. And if you don't have calculations, you can do that. But if you have calculations, that's kind of hard to do, right? So you don't have to do that. Okay? And you do need to be neat. You need to be careful, make it clear so that not just you, but the lab partner or somebody else can see it. So, why don't you I was going to have you pair up, but I think you can do it. We should pair up some, but you might have a group of three somewhere. I want you to look at your own write-up, what you did for that last lab. Compare it to what they suggest here. You notice they have references. Okay. So why don't you do that? You should pair up. Somebody wants to come over. Yeah, good. So he's not alone. That would be good. Where is? We have more than one student absent here, it looks like. I don't know where it is.
Online.
Oh, that's right. Of course. So we are all here except for one. So anyway, this is an example of a write-up. Why don't you go ahead and look through this and look what they said, and then what you did, and see any differences. And guys, your lab write-ups can be different. You can be done somewhat differently, but still meet all the requirements, and you can do good work. So why don't you take some time to talk about it, just want to compare. You may want to show each other your actual lab write-ups and see what you think and compare it, or just compare it. And then we'll talk about it. So I'm going to give you a couple minutes to do that too, 3 or 4 minutes to do that, and then we'll talk about it.
My printer is not open, so I'm not going to hit my phone.
What was that?
My printer isn't working, so I didn't get to print out mine.
You didn't bring it, or?
My printer isn't working.
Oh, yeah. I really encourage you just to do it by hand for lab reports, okay? Some of the homework assignments you can type it up if you prefer, that's fine. And I know it looks better, but you could not show your work of the calculations, and they didn't show it in this and that. If I was to grade this, I'd say you didn't show your calculations. Always want to show your work. How did they get 104 centimeters per second, and the second time 128 centimeters per second? I want to see the work, where that came from.
I thought that too.
Okay, why don't we just take some time to get you to share some things, positive, negative, what differences you notice about your lab writings compared to this one. So anyone just, what do you think? Differences you noticed? Yes.
Reference.
Okay, so some of you had included references. In the back they had references. Wiley, Dr. J.L. and Marilyn F. Darnell, that's Apologia book. If you guys use that, it would have been the same book. But whatever your book is that you use, that should, your textbook talked about perhaps something related to this. But see, this is a lab that's not really a biology lab. So if you use a different textbook, you're not going to see, if you're using Apologia, I'm sure you're going to see this in there. I don't have the book, but I assume that they do. If you use a different book, you're not going to see this lab. But you know that it's something related to velocity as distance divided by time. You can research that, you could do it online, or you could find another book that talks about it. The point was, what was the purpose of this lab? What was it even called?
I wrote it really rude.
I couldn't see it.
Like just the
I called it
working version, like,
Okay.
No.
It was a scientific method, but, and it was just,
scientific investigation.
right, it is. And that is, we want to learn how to, we want to practice in something, we're not even, this wasn't even biology, it didn't matter. What mattered was learning how to do a lab report correctly and how to go about the lab correctly. Now, this was in your lab booklet, right? So you already knew ahead of time what lab you're going to do. I'm just curious, how many of you actually read the lab ahead of time before you came to class? Okay, everyone did. That's great. That is what you should always do. Make sure you understand ahead of time. You can actually start to write things ahead of time out. They have places, tables to fill in your lab manual, and you can use it as consumable, so you can write right here if you want, etc., and do things later. But you should come prepared. You have thought through. You may want to prepare ahead of time if you're for tables to take notes, etc. So you notice the data they took. They had 3 trials in each case, and then the average did. What did I say that I thought was missing that should have been in here? I bet it was in yours. What was missing in this write-up?
The speed.
Pardon?
The speed. The
Was it the
speed.
height?
Well, the speed is, it was calculated. Pardon?
Was it the height?
No, there's something not that's on my board that was not here in this.
They didn't show all the results of
They didn't show the calculations.
it?
They got 104 centimeters per second on average. So, if you read here, it says they use 200 centimeters, which is taller than me. And their average time on this first vertical speed that they were doing without, I think, the paperclip, was the average time was 1.92 seconds. So velocity is distance divided by time, 200 centimeters divided by 1.92 seconds is 104 centimeters per second. Abbreviated seconds with S. They didn't show that work. What they did was they divided 200 by 1.92 and 200 by 1.56 to get their 2 values. You should always show any calculations you do. You won't do as many calculations in biology as you would chemistry or physics, physical science, or other labs. But you will do some calculations. And so you should always show your work that you do. It's like explaining how you got this. Magically, they said, our speed is 104 centimeters per second. And I think that's what you were saying. Where did the speed come from? So you need to show. So you should show your calculations. You don't need to show a whole lot. If you want to type up your lab report, you can do that. But you don't have to. You're making a lot of extra work for yourself. You can just do it by hand if you want, whichever is easier for you. Because in real life, that's what people do. I worked at one point. I worked for Hughes Aircraft, and later worked for Western Digital. When I worked for Western Digital, they make disk drives that go in computers, and I wrote programs to help test the printed circuit boards that are part of the disk drive. So I would take notes when I was developing a program to test it. And I would look at a board and try to figure out what's wrong with it. And I took notes of what happened each day. When I was debugging, there's different components that go on that board, and I was debugging them. I would take notes of what I did. You know, I debugged the test for this capacitor or that IC integrated circuit or whatever. But I kept notes of what I did. That helped later on to look back on it. And you did it with pencil because you make mistakes and you have changes, but it was handwritten. I don't know if you're going to type it up. And I was the only one who saw it. It was just for me, a record to help me. So you don't have to type it up, but you do want to be neat and organized. Okay. So any other thoughts or comments about lab write-ups or lab reports? Does anybody else, what do you notice that was different about this lab write-up? Because it might help others if you think about it, compared to what was in yours. Any thoughts? Anything? Did they do a better job here than you, or did you do a better job? Pardon?
They did.
They did. Okay, they did a pretty thorough job, right?
I liked I like the purpose of the experiment for my sentence. I like how I said the purpose of the experiment is to illustrate how to conduct side slaps by calculating the speed of the rover. I like that.
Okay, very good. And they did really talk about the purpose. Did you have a good conclusion? Did your conclusion look back at your hypothesis? You should have had some kind of hypothesis. And they talked about what the hypothesis is and the scientific method, but did they really use the scientific, did they come up with an hypothesis? Did you see an hypothesis in here anywhere? Guys, what was their hypothesis?
I found it. The hypothesis was correct. Oh wait, no, never mind.
That's yours or theirs?
That was theirs.
I don't think they have an hypothesis here. I don't see an
I don't
hypothesis.
see one either.
So they talked about a hypothesis, but there should have been, and it wasn't real clear the way the lab was done, how to form a hypothesis. You remember I suggested that you try to anticipate how long it will take and would it go faster or slower when you had a paperclip with a really burden. You had to think about it. Well, it's more weight, you think it would go faster because, you know, the air, because things fall at the same rate no matter what their weight, but air with paper lifts it up and it takes longer to fall. But in the whirlybird, it's like a helicopter. It's not just falling, it's kind of whirly-birding down. And the extra paperclip made it go faster, and so anyway. It did fall faster in their case. Did you guys have it falling faster when you had a paperclip?
Mm-hmm.
Okay. But that would be an hypothesis. You might have tried to anticipate how much faster. So you should always have some kind of hypothesis, just about one. Okay. Now I want to look at Introduction to the Compound Microscope video from study.com. So let me find that. Is this it? I think this is it. There it is, yes. Okay, so I'm going to go ahead and start this.
As we get older, our eyes are not able to see things as clearly, especially things that are small. We may get a magnifying glass in order to help us read and see small objects, and this helps because magnification makes the object appear bigger, so we see it more clearly.
To take notes?
A microscope does the same thing, but for very, very small objects. The most common type of microscope you'll use in your biology lab is a compound light microscope. This microscope has two lenses that bend light so that a specimen is magnified and projected. Having two lenses is very important because this is where the microscope gets its name. To compound something means to add to it, like compound interest or a chemical compound, which is a sum of multiple parts. The word microscope is also key because micro means small and scope means view. You may have noticed the word light in the name of this microscope, and that's also no accident. This type of microscope uses visible light, which is the part of the light spectrum our eyes can see, and passes it through the lenses. The lenses bend the light so that the object you're looking at appears larger than it is. This allows you to see it better. The resolution, or clarity of an image, is also an important factor when looking through a microscope. A standard compound microscope can magnify objects about 1,000 times, which is pretty good. But above this, the image becomes blurry, so even if an image appears larger, it may still not be very clear unless the resolution is good. Compound light microscopes have been around for quite some time now. They were first used in the 17th century. It's no coincidence that during the same time, cells were discovered, which led scientists to a great number of other discoveries about microorganisms and cell structure and function. Microscopes are very powerful tools indeed, but in order to use one properly, you need a basic understanding of its components. Quite possibly the most important part of the compound light microscope is, well, the light source. Okay, maybe the power switch is the most important part because that's what allows you to have light in the first place. But the light source is definitely next on the list of important parts. It's found at the base of the microscope, and you can adjust the brightness of the light by rotating the light source knob. Moving up from the light source, we find the stage. This is directly over the light source and is a flat area that holds your slide or specimen flat. On the stage, we find the aperture, which is a small hole in the stage that allows light to pass through. Whatever part of your slide or specimen is directly over the aperture is what you'll see when looking through the microscope. Also on the stage are the stage clips. These are used to hold your slide in place, and they simply clip over the edges of your slide to hold it down. There will be a knob near the stage called the stage control that moves the stage clips so that you can look at different parts of your slide without having to move it by hand. As we continue to move up from the base of the microscope, we find the objective lenses, which are attached to the nose piece. The objectives are how much the image is magnified, and the number is written on the side of each objective with the number and the letter X. For example, if you see 10x on the side of an objective, this means that the object is magnified 10 times. Most compound microscopes have 3 or 4 objectives, and they will most likely be 4 times, 10 times, 40 times, and 100 times. The nose piece adjusts so that you can turn from one objective to the other, depending on how much magnification you need for your specimen. Above the objective lenses is the body tube, which is connected to the eyepiece. The eyepiece is what you actually look through to see your specimen, and the body tube connects this to the objective lenses. The eyepiece is yet another point of magnification, so while your objectives have a magnification of 4, 10, 40, or 100 times, the total magnification of the microscope is this times the magnification of the eyepiece. Usually the eyepiece magnifies objects 10 times their size, so if you combine this with the objective lens magnification, you would have a total microscope magnification of 40 times, 100 times,
They just
400
went 10 times each one.
and 1,000 times. In order to get the best image possible, there are 2 knobs that you should be aware of. One is the coarse adjustment knob, and the other is the fine adjustment knob. The coarse adjustment knob does just what it says. It allows for broad coarse adjustment of the image. Once you get the image in about the range of focus that you need, you can use the fine adjustment knob to get it just right. The microscope also has an arm, which is the part that supports the body tube and connects it to the base. The base is simply the part of the microscope that rests on the table and supports the microscope itself. The arm and base are important because if you ever need to carry or move a microscope, these are the parts you use to do so. To safely move a microscope, simply grasp the arm with one hand while placing the other hand under the base. This will prevent you from dropping
And
or
be
breaking the
careful
microscope,
that you
which
gather
your lab
up the
instructors
cord.
will greatly
You want
appreciate.
somebody to trip on it.
No matter how large or small, a microscope is a delicate piece of equipment. It's important to use great care when using, cleaning, or moving the microscope. This will ensure that you, your lab mates, and future biology students are able to use and enjoy the microscopes for as many lab activities and experiments as possible. Named for its pair of lenses and its useful light source, the compound light microscope was first used in the 17th century. The invention of this type of magnifier led to great discoveries of cells, cell structure, and microorganisms. This type of microscope magnifies images through 2 lenses to make the specimen or image on the slide appear larger than it is. There are several different magnification settings that a microscope may have, but the total magnification of the image is always the magnification of both the eyepiece and the objective lens being used. Microscopes are very useful and powerful laboratory tools. They provide us with a fun and easy way to see things that would otherwise be unseeable. Because of this, great care needs to be taken when using a microscope. Proper knowledge of how to use, clean, and move a microscope ensures it will be around for many uses and lab activities.
All right. So you can do this, take the quiz, goes with it, etc. Okay, that's a pretty good video for us to understand the compound microscope. If you get your lab manual out, I suggest on page 3, now it has a lot of A, B, C, D, etc. You see a diagram with it, just with the eyepiece. There's what's called the body tube. Now the thing that you look through is called the eyepiece. The objective lens are the lenses that you use to magnify. It's the same thing for a telescope as with a microscope. The objective lens, the one that's closer to what you're looking at, it gathers the light. Think about it. If you magnify something 100 times, that means that same amount of light has to illuminate 100 times as much material, square area. So if you magnify a whole bunch but you don't have good light, it sure gets magnified, but it's dark, you can't see detail. And with the telescope, when I was a kid, I had kind of a cheap little reflecting telescope, and when I tried to magnify things a lot, when I'd look at some planet from a distance, it was just like a big black circle. So you didn't want to try to magnify it too much. So the two working together. So you notice if you had the eyepiece, what did they say the eyepiece is usually? Magnification? If you remember, the eyepiece you look through? It was 10, it's usually 10. And so if you had a 40 magnification of the objective lens and the eyepiece was 10, what would be the total magnification?
50.
Pardon?
50.
No, you don't have 400, you multiply. Because this already magnified at 40, and this takes the 40 and magnifies it 10 times as much. So 40 times 10 is 400. That's how that works. So, but if you look at this, we're going to basically do the lab now. Next week we're going to be doing lab 3, prepare the slide using a wet mount, and lab 4, microscope drawing. So you'd have to email me, by the way, that's what they require for this is lab report. Email me by midnight, Monday night, your lab write-up for the lab you're going to do today, which is mainly just observing and explaining it. Not a real lab as far as doing experiments to see what happens. And then you have a hard copy you'll bring to me next week, next Tuesday. And that's and by the way, the one we just did, which we gave you the example. So what you should do is go ahead and make any changes you want to in yours, and you can bring that in next week. That's good. So both of those could come in. So why don't we get we have 3 groups, and the microscopes are down here.